Gar. And only 20% of these stories go on air in TV, around 40% go on air in, go on print in web. The rest, we can't process it. This is verified information that comes, but we don't have enough hands to process it, so we don't. That's where a crucial development that's happening in US or UK is catching our attention. What Washington Post is doing, it now has bots they're writing local hyper news. Bots are doing the job. You don't need a person paid salary to work on it. So that's where we are looking at some sort of innovation from the tech industry. If we have bots which can just put out an article, 150, 200 word article, taking out information, verified information from what our reporters, stringers from across districts in India send, then we are looking at those 300 stories also coming on print or somewhere in the apps. And also the fact that there has been so much of analytics used in news as well. What News UK is doing, that's also an example. They are looking at habits, reading trends, every aspect of a reader's choice, and then tailoring news for them. That's something that we are looking at that will help our apps in a big way. The second aspect that has been spoken about in the last couple of years in India is fake news. Facebook might have demoted news articles in a big way on their feed, but we Indians haven't stopped sharing fake news. So that's where fact checkers come into play. We have BoomFact, FactCheck, and various such organizations. But in India, what's happening is every story is manually verified by their reporters. They call up, ask if this is true. But there's a development that's happening in US and UK, which is there's a portal called Nowhere News. I was just checking how they work. And they have 1,000 verified news sources. So every news that comes in, they cross-verify with these 1,000 odd sources and then put out a very non-biased, actually just facts, no views, and then they verify the story. That's something that could play a huge role in India, considering what's happened due to fake news in India. The other aspect that is of a worry in a newsroom or for the editorial is that we've seen a lot of analytics used on our websites and various other portals. At the same time, many believe this analytics will lead to formation of an echo chamber, because usually tailoring of news means they will only cater news that you like, and at times the other side doesn't really get the story there. In a news channel, you have everything being played out. What Mr. Modi says, what Mr. Rahul Gandhi says, but if you look at the Facebook feed or some of the news articles, they will just send out stories. If the pattern comes out that you like news about the BJP or the union government, then you will have such stories. Or if you're someone who's reading a lot about Congress, then such news stories are pushed. So that's the worry for the editorial right now, that we might end up having an echo chamber. So a lot of people from tech here, we're hoping that when you design your products next, you can keep these needs of the media industry in mind. Thank you.
Thank you, Mr. Upadhyay. Moving ahead, we have with us Professor Ramaswamy, Institute for Stem Cell and Regenerative Medicine, Dr. Shambhavi Naik from Takshashila Institution, Dr. Rohan Kamat from Viravecs Labs, and Vijay Chandru from Strand Life Sciences to discuss the past, present, and future trajectory of gene editing. Thank you.
So very happy to be here, and you know, I think on this whole issue of gene editing, for those of you who opened the Mint today, you would have seen a very nice opinion piece by Shruti and others from the Carnegie Endowment. So you know, I would urge you to go look at that. Today, we are going to discuss the power of gene editing, and of course, in many ways, this is really a little bit about both the present and the future, but we will get a wide view from the panelists here. I'm just going to set the stage with a few comments, and then we'll move it forward. So, of course, this idea of gene editing has been around for a while, but in particular, once we had finished writing the human genome and other genomes, I think the whole challenge sort of turned towards, we have finished reading, we have now turned towards writing in the genome. So I think the quote from Eric Lander sort of indicative of that. The financial implications of this technology are starting to really be felt. If you look at various private equity projections, these are put at somewhere between $4 and $5 trillion just in human health because of the ability now to think about cures using these new technologies. I think, but it's also important to realize that this is not only about human health, it also goes into aspects of working on other organisms, plants, microbes, etc. So I think our panel will also sort of focus on that. The idea of gene therapy has actually been around for a long time, almost 40 years. And there were various safety issues, and things were sent back to the bench, and now it's again out on the bedside after some of those issues have been corrected. But there's basically two ways in which this type of gene therapy is done today. One is where you autologous, so you actually draw cells out of the patient, do some editing on it, and then put the cells back, and then the patient improves, or the disease condition abates. The other is what is called allogenic, where you could actually collect cells from a donor and then insert them into the patient. So there are various aspects. We are still mostly in the autologous phase. I think everyone's looking forward to the allogenic phase because that would really change the supply chain and for large-scale sort of implementation of these technologies. There is a problem with the way in which DNA is delivered through viral vectors, and I think one of our panelists is actually a specialist on that, but we'll deal with that later. You already heard quite a bit about CAR-T. I think yesterday Dr. Siddharth Mukherjee had spoken about this. I think the two big approvals that have hit the market are, of course, the Novartis and the Gilead therapies. I think this has largely been focused on hematological disorders, but everyone is looking forward to a broader sort of application. CRISPR-Cas9 is the cut and paste technology, which is really the exciting way in which you could deliver, instead of using the viral vectors, you could actually go in and actually edit with a cut and paste technology, and there are many, many developments around this. I just want to sort of set the stage again by pointing out that there are ethical dilemmas here, and Shambhavi will probably speak to some of them. But on the one hand, there's inequality issues because of the cost of some of these therapies, but it's not just for children. There's also adults who are waiting, and For those of you who follow sort of genetic disorders in India, we probably have somewhere on the order of maybe 50 to 70 million, depending on how you estimate, of what are called rare diseases. Of those, at least half are strong Mendelian disorders. So we actually have a huge population that could look forward to some of these therapies being helpful to them. I just want to point out that a lot of people have sort of read about this Chinese announcement where there was editing of an embryo, and that has created, in fact, Shruti's opinion piece talks primarily about that. But here is also at the bottom there, the picture is of Dr. Amato from the Oregon Health Sciences University, and she has been working on editing the embryo. But she's been doing it for the myosin-binding protein for cardiac type 3, right? Which is actually a genetic marker that causes hypertrophic cardiomyopathy, which is a deadly disease, and it actually affects a lot of South Asians. So, is it a good thing that someone is working on it? Or because, there is probably a cure that's going to come from that. And so these ethical dilemmas exist, should we work on these or not? So with that, I think maybe I'll just ask the panelists to make some remarks, and then we can open it up for questions from the audience, please. So maybe we'll start with Rams, because he'll tell us about sort of the basic biology research, which is driving this whole technology, and then Rohan, who will talk to us about sort of some industry perspectives, and then Shambhavi on policy. So please go ahead, wherever you like. I think we are mic'd up, so you can.
Maybe just sit here and do. Okay. So that way we can keep it short. I'm not going to tell you what the technology is, because Chandru already introduced that to you. I come from a very academic perspective. I'm a scientist at the Institute for Stem Cell Biology and Regenerative Medicine. Often, the scenario and the announcements that was written about today gives you this Frankenstein kind of an idea that human beings are beginning to play God. I'm going to step back and say, what do we do in the lab, and where do we use these kinds of technologies from? Normally, when you have a discovery like this, it first happens, it goes to the laboratory where people are fiddling around with things. So I'm going to give you an example of not necessarily therapeutic or agricultural inventions that you will see, but what we do in the lab and how it could impact you, and I'm going to give you an example. So the Stem Cell Institute, we work on a little worm as a model system to understand regeneration. And it is well known that there are genetic mutations that cause degenerative eye diseases, okay? And we wanted to understand what a specific mutation that is a developmental disorder that causes formation problems in your eye when you grow old, and how they do things. It turns out there is a mutation in a particular gene that causes this disease. And one could argue that, oh, we have this CRISPR-Cas system, we're going to engineer this gene, put the cell, and make the person okay. But we need to understand how this disease works. So we took this little worm, and it also has an eye, and it also has this development. And you knock out this gene, and you begin to study what is happening during the developmental process of the formation of the eye. And it turned out that it affects making a small compound called serotonin at a particular stage during development. And serotonin is a neurotransmitter, but in this case, it is playing a different role of telling cells, you go become eyes. Now, if this gene didn't work correctly, there is no serotonin produced at the time, and there was no signal for your cells to become eyes. Now, what have we done? By understanding how this works, I actually don't need to do any engineering. If I could fast forward into the future and say, you know what, if you have this gene mutation, I don't need to do any gene therapy. I could just have serotonin at the correct developmental stages. I could just give you a drug of serotonin. So most of the time, what we are doing is using these types of technologies to understand how a disease is caused, and then ask, if this understanding is this, how can we later intervene? I mean, that's a later process. I think therapy is much later. So this allows us to model diseases. This is one thing. The second aspect is, this year's Nobel Prize went for a person called Francis Arnold in chemistry for doing directed evolution. What she did was, oh, I want to make bacteria do something. And And we all know about evolution. So if I'm going to go to pressure on it, it's going to make genomic changes and evolve the bacteria to do something very specific. This is several generations. Can we use this kind of technology to do something slightly different, where I can actually specifically engineer a bacteria to do something that I want it to do? What am I going to use it for? Let me give an example. One of the biggest uses of petroleum today is making nitrogen for agriculture. 70% of you and me are petroleum because all the nitrogen comes from the Haber-Bosch process. Can I now engineer better bugs so that I don't have to use petroleum-based fertilizers to grow my agriculture, instead have the Haber-Bosch process happen in a reactor in a cell that I could use in agriculture? So it's always not necessarily a genetically modified crop or a Frankenstein person who was created, but there are many interesting ways to use these technologies. So in the laboratory setting, we are using this for understanding and seeing how else can we think about things differently. I mean, that's the primary focus. Sure, I work at the Stem Cell Institute. We are definitely interested in using these kinds of engineering to modify stem cells, but that's a long-term goal. Let's not forget that it's understanding that allows us to manipulate things. And a lot of these tools also help us understanding, and I'll stop there.
Thank you, Rams. Yeah. So, Rohan?
That's actually very nice how Rams has put it, that a lot of study has to go and is going on in about how genome editing is going to benefit the society in, say, therapies, crop production. We too work in the, in the human disease sector where we develop different tools for gene therapy. But time and again, we get calls from various seed companies, agri companies, saying that we need to do some tinkering around and can you help us? So typically I ask the questions, what about the licenses, approvals to them, because I don't come from the sector, but I would like to know about their stance on it. And unfortunately, a lot of them say that we'll look about that later. But can you help us? So that's where we've discovered that now in a research lab of his or mine, if I have to do even tinkering on cells which are in a glass plate, we have to have a bunch of approvals. Whereas now we are talking of plant companies which are doing tissue culture, which are seed companies which are making seeds. And I ask them about approvals and they say, you know what, don't worry about it. That's something that is manageable. And that's something that has scared us. And the repeated inquiry which comes to us, we don't go in that sector. That sort of has put us in a big questionable situation. What is the government doing, or what are the scrutinies that have to be done on these kind of experiments that have been done by commercial entities? And that's a concern that I would like to put forward in this discussion. But otherwise, yeah, I mean, the way things are moving in terms of therapeutics, it's really good. I think in the next couple of years, we will see some therapies. Editas, I think, has gotten the first FDA approval for CRISPR-based therapies. So let's see, hopefully in a couple of years, we should see something. And CAR-T, which was a viral vector-based therapy, which you heard yesterday, took close to 10 years to come to the market, but that was also because the science was a little slower then. Now the way technology has caught up, we will see therapies with using editing quite soon, I believe, in the market. So, yep, that's my opinion about the whole thing. Over to.
Thanks, Rohan. So, Shambhavi, now I think Rohan has actually set up the stage for policy wonks like you to,
yes,
right?
So I must say, I work with Takshashila, and I'm a policy analyst who works on science policies. And gene editing is an extremely exciting challenge for a policy analyst because of the varied applications that it has. The first challenge is that the narrative actually puts gene editing into buckets. So we say GMOs are bad, we will say gene editing is unethical. Whereas as a policy analyst, I have to actually distinguish between each application and judge each application on its merit, because it has different risks, it has different benefits, and some might actually be beneficial applications. So I have to take a stance back and say that I can't put things into a bucket. So once I start looking at applications differently, then the question is how to balance them, right? We want to promote the kind of research that Rams does in his lab, but we would probably want to regulate the kind of seed companies that Rohan works with. So how do we actually achieve that kind of balance? There are good applications of gene editing. Precision medicine is probably something that we would like to promote, but at the same time, bioweapons is probably something we would like to prevent, right? Biohackers, DIY CRISPR kits. So how do we actually achieve that kind of balance when we work with dual-use technologies? The third is who achieves this balance, right? Who regulates? What role do institutions have to play in shaping that narrative as well as in regulation? And the fourth is what role does the public have to play in this discourse? And some technologies which will impinge on public life, I think public discourse is extremely important. So how do we include that in policy making? And the fifth problem is how do we integrate technology with society? The example I would like to give here is of Green Revolution. You probably all heard of Green Revolution. Technological success, yes. But one of the most important factors of why Green Revolution worked is the minimum support price. The idea of Green Revolution was to increase supply of food products, right? Increased supply by simple economics means lowered prices. The moment prices would have crashed, farmers would have suffered. And that's why MSP was brought in to ensure that there was an incentive for farmers to continue farming with the wheat and rice varieties that were coming in. How do these new technologies coming in, how will they impinge on society? We don't really know. How do we forecast it? Do we have mechanisms to forecast it? Is primarily a very important question. If we talk about precision medicine, we talk directly about, like Tom said, about these super soldiers and designer kits. But if it does work, if people live longer, what questions will prop up then? Can we actually provide a living for them? Can we, if gene CRISPR actually happens, what kind of society we will live in? This is questions that as a policy analyst I have to think of right now before I take a stance of promoting a technology. So this, I mean, it's an incredible science to deal with, but its integration with society is going to be very, very difficult to think about.
So thank you, Shambhavi. But maybe I can ask a couple of questions and then we'll open it up for general discussion. So Shambhavi, you and your colleagues have actually drafted a set of guidelines, right, for the regulation of gene therapy and gene editing, which you presented at CMC Vellore earlier this year. What is the status of the document, and is the government taking it seriously?
Let me just start with where we started off with this document. We had a visitor from outside India who came and said about how gene editing should be banned worldwide. And we started a discussion about whether it is in India's interest to have gene editing applications. And clearly this is not for a small group to come up with. So the way we thought we would do this is come up with governance frameworks instead of having like a strict rule of guidelines. The problem with emerging technologies is that laws don't work because they're very rigid, they're inflexible. So how do we actually, if there's a law today that bans something, how do we overturn it? So guidelines are probably a better way to go for it. Unfortunately, they are not enforceable then. So what we suggest is that we put in standards, accreditations that labs can follow. The way we went around is by studying what other systems have used and then had a lot of stakeholder meetings. I know Dr. Chandru was part of it, Rams was part of it. We spoke to patient group representatives, lawyers, ethicists, so a wide variety of people to come up with these governance frameworks as tools that can be used to look at gene editing applications. We also try to tackle the challenges that I spoke of earlier. So what the framework suggests is that we create specialized working groups for each and every application instead of bucketing into one policy fits all, because I don't think that will work. So for example, if you want to look at gene editing for thalassemia, which is already under observation elsewhere, then you create a group of doctors who work with thalassemia, of scientists who work on thalassemia, of patient group representatives, of patients, lawyers, and they come up with a guideline of what is acceptable, what is not. It will take a bit of work, but I think that is the only way gene editing can actually go ahead in a more inclusive manner. So we have been talking to scientists across India, like Dr. Chandru said. We spoke about at CMC Vellore. We wrote a piece in Nature India a couple of weeks ago, publishing the frameworks, and we have been trying to get into the government as much as possible. So we're working with Dr. Chandru on that, but yeah, hopefully we do need guidelines. Either way, whatever India decides to do, we do need gene editing guidelines, and urgently.
And going on, you've also written about gene drives, and maybe you could explain what gene drives are, and then what the variety of applications they go to, and and some of the issues there, right?
Can I just quickly take a poll of how many of you have heard of gene drives? Okay, one hand. Genetically modified mosquitoes.
Have you
Okay,
heard
few
of
more hands.
mosquitoes? Yeah, lots of people have
Okay.
heard of mosquitoes.
So this is
right?
one of the problems of gene drives that we were discussing earlier, is that this is this bucketing problem. All gene drives are genetically modified mosquitoes. No, gene drives have other applications, but I have written about genetically modified mosquitoes in detail. And the reason I've written is because I don't think that we have given enough attention to the topic as much as it deserves. The Chinese experiment scares us, but we kind of have neglected gene drives a bit. So let me quickly explain what it is, okay? And bear with me for a couple of minutes. I'm simplifying science to a degree that it might not remain science, but this is just for sake of explanation. Please don't take it back with you, right? So how does genetics normally work? So suppose I inherit 2 pencils from each of my parents, right? My dad gives me a black pencil, my mom gives me a white pencil, okay? So I have 2 pencils. Now, when I have children, I give 1 pencil each, right? There's a 50% chance that my first child gets a black pencil, there's a 50% chance that my second child gets a white pencil, right? So that black pencil that has come from my father is with me, and then it goes to my 1 child. When they have 2 children each, 1 out of those 4 children will have that black pencil, right? So 1 in 4 chance that that black pencil goes from my father to my grandchild, right? 1 in 4 chance. This is how genetics routinely works. There's a 50% chance of me passing on my genes to my kid. Gene drives is taking this a bit further. It's a natural mechanism by which some genes have managed to corrupt the system and pass on 100% times to the offspring, right? So in that case, if my black pencil was a gene drive, all 4 grandchildren would likely have the black pencil, okay? So this is how gene drive is different. What it means is that the gene that I have will spread rapidly in the population, right? As opposed to other genes. This is naturally occurring. It was first discovered in flies, in fruit flies. And the narrative at the moment is that it is being used for vector control, right? So mosquitoes, to control mosquitoes from spreading malaria, dengue, etc. It is still a nascent technology, and there are scientific questions that remain to be answered, and they are being answered. And they should be. I mean, if it works, it is a great way of preventing infectious diseases. But at the same time, this technology differs a bit from other gene editing applications. If, as a consumer, I believe that GMOs are bad, I have a choice of not buying GMO food. If I believe gene editing is unethical, I have a choice of not getting my child genetically edited, right? But if my government decides that it will deploy genetically modified mosquitoes as a citizen, I really have no other choice apart from moving, which is why public discourse is extremely important in this field. At a later stage, not currently. Currently, we are still in the research state, but once it comes to deployment, public education and public consent, I have no idea how we take public consent at all in this case, but it is going to be extremely crucial. How many of you know that there have been trials of gene drives in India? Field trials.
Nobody.
Couple of people know. So in Maharashtra, this British company called Oxitec has been doing closed cage trials. So this is what I'm trying to say, that once it comes to the stage of where this technology comes in contact with public, we need a wider discourse to happen. Which is why I think gene editing has a lot of policy issues which are different from other gene editing applications.
I think it's also important to understand that gene drives, I mean, this mosquito application is, of course,
a single application.
yeah, single application. It also applies to other situations. Maybe Rams, you want to?
No, so it applies to many things, right? So essentially it is anything that is modified and let into a population. We have no more control of it either. So if I decide, for example, to make a new nitrogen fixing bacteria using gene drive, that all of the bacteria hereafter, all of this fungus hereafter will have this gene drive and make better, more nitrogen. There's no way I can take it back.
Yeah,
it has gone off into public.
There is no recall at all.
There is no recall possible. It's not like a drug. When I see a new side effect, I say you can't sell this drug anymore. It's become part of the ecosystem. So it's an interesting challenge.
Right. So on that note, Rohan, how is gene editing of plants and seeds different from GMO, right? And how does this affect how regulators should approach it, right?
So today morning, Rams made me aware of something, and what a couple of weeks back was that, where European Union has classified gene-edited crops also as GMOs. Now, GMOs are typically crops where you've inserted a foreign gene to typically give disease resistance. Gene editing, on the other hand, is where you tinker on the genome. You don't insert any foreign piece of DNA material in there, but sort of do a little bit of cut, paste back. There's no foreign material going in. You just break something and the cell patches it back itself. And by that, you can sort of change the gene function. So that's the biological difference. But now, when the process happens in cells, the cells do it according to their way, not according to my way. Okay, and that's where the editing may have side effects. One of the effector parts of the whole CRISPR pathway may have off-target effects as it is called. And that is something that has to be studied. So now comparing with GMOs, GMOs are where there's a foreign gene in there. Gene edited, there's no foreign gene, but you've done some bit of tinkering in there. European Union has put all of it together, and think about it, the GMOs typically take 10 to 13 odd years to come to market, and millions of dollars. So now what is going to happen is that if somebody has figured out a way to make a good, say a crop variety, say take a better apple which is much resistant to cold weather using editing, that has to go through the whole GMO pathway cycle with multiple trials before it hits the market. And now mixing both of them may not be the best option necessarily, because you're pushing tech back, and it's going to take a long time till it's the need. I mean, look at India. I mean, considering our population, we really need something that makes our crops more stable, the product produces higher, maybe quicker. That's the need. I mean, so but now they are mixed by EU. We don't know what India has as stance. I mean, I'm not.
So I think to summarize quickly, I think basically what you're saying is that gene editing of plants and plant varieties should probably be viewed in a slightly different way.
Yes.
And as Shambhavi had pointed out, maybe we need subcommittees that actually deal with these regulatory issues. Rams, in genomics for disease modeling, right? I pointed out the myosin binding protein for hypertrophic cardiomyopathy, and people at NCBS in your faculty actually work on this and are working towards perhaps finding therapies, right? So could you elaborate on some of these exciting opportunities, which are very South Asia specific, so very important for our own populations, right?
So many of these kind of genetic diseases we know of mutations are from studies that have been done outside of India. And often those mutations don't directly reflect to our population, because we are diverged from the original species very long ago. So what Dandapani at the Stem Cell Institute has done is he worked with the same person who we talked about in Oregon Health Science, and has cataloged the Indian mutations that, for example, cause cardiac hypertrophy. Now, we know there are these specific genes that are causing cardiac hypertrophy, and he also studies what type of phenotypic effect and when you can get this disease. So you can actually begin to know, if I know you have this mutation and you are a South Asian, you use more rice, you eat more wheat, I can begin to proactively intervene at an early stage so that you don't get the disease, or postpone the disease occurrence. You can't stop the disease because it's germline mutation. So that is the cardiac hypertrophy. So there is also interesting studies that we're doing with NIMHANS, for example, between patients who go from have depression, who get to dementia, and who get to other neurodegenerative diseases. Can we study families and with specific mutations in the Indian cohort that go from depression to dementia? So again, these are all diseases that are just not genetic, they are also environmental factors that involve. So understanding at an early stage that you're prone to get depression, can you then provide them with an environment that can postpone the occurrence of disease? And these are more important as we're beginning to live longer, both cardiac diseases and neurodegenerative diseases, as well as cancer and stuff like that. I'll just give you just two examples. So it's a very exciting time that we can understand this at that level.
Absolutely. And I think, I should have sort of started the session by talking about the fact that, this revolution in genomics is actually very exciting. I think many of you were enthralled by the information technology buzz that was happening before us, before we came on the panel here. But genomics as well has entered what we call the second half of the chessboard. So the fact that we can, what that means is that the cost and the speed and the ability of sequencing DNA has gone up by a factor of 2 raised to the power 32, or about 4 billion times. The technology improvement can be measured, right? So essentially what, the ability for us to sequence genomes, annotate them, interpret what they mean, has now really accelerated, right? And because of that, many of the things that we are talking about are now possible. And I just want to mention that earlier this week, there were discussions between the Indian government and the Earth Biogenome Project, which is actually seeking to catalog the entire biodiversity of the Earth, right? And because now sequencing technologies have reached a certain point, we can go beyond just creating databases which are just catalogs of species and taxa and so on. We can now add genome sequences to go with those. We can barcode specific animals. Today's newspaper says Kerala has barcoded the DNA barcoded all the elephants held in captivity. So now there's a unique identifier associated with each of the elephants. So we can actually do this now for many organisms and actually build a very rich database from which all kinds of applications can flow. So it's a very exciting time. I think we have about 12 and a half minutes or so left. I'd like to open it up for audience discussion, so please. There's an arm raised. Could you please get a mic to the person? Right, thank you.
Thank you very much. My name is Marion, Professor CMR, University here in Bangalore. And I have a question for Madam Shambhavi. Shambhavi, thank you. In 2002, Michael Crichton wrote a very interesting book about gene editing. And the drama was that it was not possible for the very passionate team regarding the game gene editing to anticipate on the consequences. Consequences in society, industry, and our human species. Now, secondly, in the book, it's a novel of course, there was no benchmark. They didn't know how to create rules for government or industry, etc. because they hadn't a benchmark. Now my question to you is, because it's indeed an exciting time, but it's also very innovative and you're stepping in the unknown of the unknown. Yeah. Do you have any benchmark to create for society, some game rules, and the industry, of course? Thank you.
So that's a brilliant question, because that's, I think, what all policymakers grapple with even now, is what do we send the benchmark to? And I am probably not the correct person to answer that question, because I can't speak for a nation, or I can't speak for the technology itself. What I have understood from working on this field, though, is that India needs to set its own benchmark and not look outside. Our problems are very unique. Our genetics is very unique, and we need to be looking internally for discussions. We cannot be looking outside. One of the things that have come out from the experiment that was held in China is whether it's ethical, whether it is not. And the question really is it ethical for India to be doing this? And when we have that discussion, we have to take into cognizance the fact that our healthcare systems are not as strong as those of the West. Our standard of living is not as good. Simple things that our schools are not wheelchair friendly, right? So the pain of a genetic disorder is multiplied very highly when it comes to an Indian citizen. So if we can have technologies that can prevent that, should we really be thinking about what the international alignment is? We are part of a lot of alliances, which means that we have to take that into hand, but we have to now step up and think that what is our benchmark, what kind of society does India want to have in the future, and what kind of healthcare do we want to give. Now this is only from a healthcare perspective. It can similarly be applied to say GMOs or gene edited plants and to all the other features, right? Do we have that? No. What I suggest is that we create frameworks because that benchmark also needs to move from time to time. I cannot set a benchmark today and say this is what India is to follow for the next 10 years because tomorrow there might be a different discovery and change everything. So what we are trying to do at Takshashila is create those frameworks instead of saying that this is your set guidelines and this is what you follow. And then hopefully that those frameworks can guide people to take correct decisions. I hope that answers your question.
Yeah, thank you. And perhaps, yeah, we can take at least 2-3 more. So please go ahead. Right. And one at the back.
Thank you. You know, the impression I got listening to something that Rohan said was that perhaps the seed companies who are working in this field are sort of putting aside or not really observing the kind of framework which is being applied to, let's say, your work. That's the impression I gathered. But I thought, I mean, in that sense, is it because the 2017 December guidelines issued by the Department of Biotechnology, are they not equally applicable? I thought they were equally applicable whether it's related to plants, animals, and human health.
Yeah. So they are applicable. The problem is about the scrutiny and the follow-up on them. Here when, and again, I'm not talking about all seed companies or all plant companies, bunch of people who've talked to us, a handful of them. What happens is the kind of governance that is there on when it comes to human application, it's much, much more as compared to what is to do with crops. I always say something that, you know, here when we talk about plants and crops, you're talking about edible plants. But what about editing ornamental crops? Okay, flowers, something. But that may not be food for us, but then that is changing the ecosystem, right? So, and who controls it? I don't know if there are any plant tissue culture companies here, but I have got a feedback from a lot of them when I ask them how many times are you visited by any government body to keep a check on what you guys are doing. They say, well, never. And that's where the concern is that now if I'm a rogue scientist and I decide I don't care, I just want to do and make more money. And that's where the trouble is, where for us, every year we have to get through approvals and sanctions to go to the next year. And I feel that when you mention the rules are the same, the governance, the bodies overlooking it may be something that we need to work on.
So again, you know, the whole issue of implementation and actually, you know, regulation on the ground, which I think has always been a challenge. We have the regulations on paper, but how we actually implement it.
Vijay Chandru, can I also?
I think,
Can
yeah,
I just
please,
add, and
quickly,
correct me if
please,
I'm wrong,
so we can take a couple more. Yeah.
Our guidelines are not enforceable. They're not legally binding. And they pertain to funding. So while Rohan's lab and others are funded by the government, private entities are actually allowed to do what they want, right? And that is the main problem of the guidelines. Yes.
No, I think Rakesh wants to clarify that.
No, because as far as I know, let's say if you are working on certain technologies, then your lab has to have certain clearances and et cetera. So, and if you don't, then there is a criminal case. So it is a criminal offense if you are actually, yes?
No, that is only for genetically modifying, not for genome engineering.
Not for genome
So there's the clarity.
That's exactly
Essentially what you're saying is, if I take a foreign gene and put it on a plant, I need to do that. Genome engineering, I'm not putting a foreign gene, so it's only guidelines and not.
Guidelines.
I think there was an earlier question.
My question is to Mr. Ramaswamy. There's an alarming trend of increase in diabetes and cancer. Any progress being done on that as of now? And for example, polio and smallpox has almost been eradicated. Is there any deep thought process being done about this?
Well, I think, you know, we did mention in the outset that there is now a lot of excitement about CAR-T, which is basically
Cancer.
directed at cancer. And
So I'll tell you about diabetes. I was going to let
Right.
Chandru answer cancer, and then
Right.
I will do the diabetes bit, right? Yes, there are interesting ways by which today we can take your damaged islet cells and reprogram them to remake insulin. But these are all things that we can do in the lab. By the time it reaches a patient, it's going to be 10 to 15 years. But it's an exciting time where if we can actually induce regeneration of your islets of Langerhans, I can, in fact, fix your own body to fix its own diabetes. And these are the things that are happening. Nothing on the market yet, but exciting times.
it's also true that diabetes is a complex genetic
yeah.
disorder.
Yeah.
So, the disorders that are Mendelian, so there's a very strong penetrant genetic trait, can be attacked first because those, you know where to go and what to do. Whereas, complex disorders have a little more of a challenge.
When you mention about polio, that's single virus. It's a single
Very
entity
easy,
causing
yeah.
the disease, as opposed to being a multifactorial disease.
Multifactorial
So
disease. Yeah.
that's where the difference
And
is. when it comes
diabetes
to-
is,
And
unfortunately.
so is cancer.
So is cancer.
And so is cancer.
It's very difficult. Yeah.
So,
Many
we'll
questions.
take maybe 2 more questions, please. Whoever was, right.
Sorry?
Hemophilia, right.
Oh, yeah. So, yes,
Yeah.
there is a full-blown study being done on hemophilia. That might be the first
It'll be the first
clinical trial that
yeah,
might happen in India. It's gone to 1 round of approval for clinical trial to the CDCSO. The second round, the responses have been made by CMC Vellore and the Stem Cell Institute. Hopefully, once they accept the responses,
So the
clinical trial will start.
InStem at NCBS has a translational center
At Vellore.
at CMC Vellore. And CMC Vellore is likely to be the first site of a clinical trial for hemophilia using gene therapy, which is, I think the applications are into the regulators. So I'm on the advisory board. So we're actually looking forward to the trial getting started. It'll probably happen
Very
sometime in 2019, hopefully early, right? So,
Hi. The question, any examples of applications of artificial intelligence or machine learning in this space?
well, I'm basically a computer scientist.
Yeah.
And when we got started in genomics, the first thing we did was we built machine learning platforms for being able to analyze genomics. And little beknown to the public here, I guess, Strand was the runner-up of the KDD Cup in 2006. So, yeah, I mean, this replete with applications of analytics of various kinds. I wouldn't single out AI per se, but analytics of various kinds.
So I just want to add one thing to that. I mean, the genome revolution itself is very hard for us to comprehend, and we are still waiting for good math and good software and good AI and good whatever to do this, right? What is more complex is what I told you. I find a gene mutation that causes a change in a metabolite. I could find a gene mutation that could cause a change in a protein or a post-translational modification of a protein. We are just beginning to unravel this multidimensional stuff that the biology is doing. And I think the late limiting step in our understanding is actually not going to be the tools to generate this data, but to put this data together.
Right. I'm afraid we've run out of time. And we actually have a minute.
Yeah,
maybe we can take one more question, please.
I want to get your assessment of how you think that overcoming thousands of years of evolution would actually be easy to do. You're unleashing things out there that ought to survive and overcome things that have actually are proven over thousands of years of evolution.
Sure.
What would you think it would take to actually have a realistic assessment of what would actually survive the evolutionary trends and so on, what we'd be able to overcome them?
Well, I don't think we have good predictive models of to be able to make statements like that. But the crack in creation, as Jennifer Doudna calls it, the crack in creation has been created, and it's wide open today, right? So the question is, you know, not whether it's going to happen. It is happening. We are tampering with genes. And what impact it will have is partly for evolutionary biologists and ecologists to you know, keep working on, because I think this is going to happen. Now, genetic modification at some level has been happening anyway, right? Yeah. When we do breeding, when, you know, there's a lot of selection that happens. And we have, you know, in some sense, we are in the Anthropocene period, not just because of climate change, but because of all kinds of things that humans have intervened with nature with, right? And therefore, I think, you know, some experiments have been carried out. We've seen the impact of that. Others will happen, and they'll happen at a much more rapid pace. But you're absolutely right. There should be an agenda of research to understand what the implications are for the Earth's ecosystem. With that, maybe we'll have to close, but we can continue.
Can I make one comment? I think in the end, because we know what is the positive impact of genome editing, we need to remember George Church's statement that we have an obligation to society to be balanced, whether we are policymakers or scientists in any field.
Sure.
I think we definitely have an obligation to be balanced.
Right. You should talk to Binay. He's part of George's team, so he's right here. Right. Thank you very much. Sorry for delaying your lunch, but I think hopefully you got something out of it. Thank you.
Interesting discussion. We now break out for lunch, and lunch is served right outside the venue. We request you to please join us for our next session on building a global cybersecurity landscape at 1:45 p.m. Thank you.